assembly directory, and I've highlighted the context.fasta. That's the first one we want to take a look at. Yeah, as we said before, the first way to start exploring the file is just doing the head, the tail. Maybe if it's small enough, you want to vim or nano it, yeah, so that you can take a look at the file and get a feel of what it contains. From this point on, if you remember and if you're familiar with the Fasta format, you should know what to expect. A Fasta format is just another way of showing sequences. This is not a FASTQ format like the one you get from sequencing machines, where you have one line for sequences and another line for qualities. No, this is a FASTA format, which means that you only have sequences stored here. There is no quality associated with them. The file is small enough that we can beam it, and we can explore a little bit what it looks like. It looks okay, I guess. It's just a sequence. There's nothing we can really do with this. Yeah, at this point. We're just taking a look at it. Yeah, and we're trying to find a way, hey, how can we take a look at this file, yeah, and get some meaning out of it? The first thing is knowing or understanding what a FASTA file is, yeah, and how it is organized. If you noticed in the first line is not a sequence. The first line, it's a name. And there's a characteristic about this name. It starts with a greater than sign. Have you seen it? Are you all there? Yeah, please take a look at your screens. Yeah, and you'll see that the first line, it starts with a greater than sign. That greater than sign, yeah, it's what is going to separate one sequence from another inside a FASTA format. Once a sequence finishes, the next line is going to be, again, a greater than sign with a name and then another sequence. That's the FASTA format. So, in order to kind of summarize this a little bit, yeah, let's see, how many sequences in total do you think there are? Should there be one sequence per name? Should each of these sequences, all of them, have one name? A different name, I mean? It makes sense, right? That all of the sequences are stored in the FASTA format, each of them will have its own associated name. And all of those names will start with the greater than sign. So that's what we're going to look for. We're going to use one of the most used commands in Linux. It's called grep. G-R-E-P. If you want to find the occurrence of a word, of an expression inside of your file, yeah, grep will help you. Don't push enter yet. I want to explain what we're doing. So basically what we're saying at this point is grep is get regular expression, or obtain, capture the regular expression. But what? What regular expression? What we're doing between quotes is saying greater than. Because we said that in FASTA format, when we do the greater than sign signals that there is a name. So what we're going to do is capture all the lines that contain that symbol, the greater than.
Make sure you put that greater sign
between quotes. Between quotes, all right? Grep, greater sign. And that greater sign should be between quotes. And then we tell them, hey, where should you look for that greater sign, the greater than sign? In my file context, oh, sorry, there should be a space there, context.fasta. Now you can press enter. In my case, it's not going to be the same for everybody, but in my case, I only got two results, two names. Node 1, length something, coverage something. Node 2, length something, coverage something, right? Did anybody have any results that is different than 2? More than 2 lines as output? Or did anybody got just 1 line as output? Basically, this is telling you how many sequences you have in your file. Because you know that there are this many names. Yeah, and just because of the name, just because this line tells us, you know that node 1, which is your sequence 1, will have a length of 111,329 base pairs, and it has an average coverage of 46.15. That's in my case. Each of you is going to have a different name with a different coverage and different length, right? But this is just because the assembler is a good one, and it's giving us good information or decent information about what are the characteristics of each of these contigs. Yeah? But what if we didn't have those characteristics? How do we explore a sequence without knowing if the names don't actually help us? What if the assembler had named them just contig 1 and contig 2? And that's it, no further information in the name. It is possible. Some assemblers do that. Yeah? How do we know basic statistics about what is inside my sequences? Yeah? There is a kind of quick and dirty way, which is the one we're going to learn right now. And there are fancier programs that can actually estimate all the parameters that you want in a single command. Yeah, but it's better if we know and we understand where each of those metrics come from, what each of those metrics mean. Because each of those metrics tells us something about how good our assembly is. Yes? So we have finished the assembly. We know what's the length of each of the contigs because of that, but let's assume that we don't. Then we have one of the most used tools in bioinformatics. For pretty much everything you want to do with sequences, you're going to end up using these tools. The tools is called SUM tools. I'm pretty sure that if you've read any papers about bioinformatics, assembly, etc., you must have run into the name SUM tools. It's written like this. If you press enter after this, all of you guys who have BioLinux, yeah, you should get a help. SUM tools is a tool meant to analyze pretty much everything that is associated with genomes. It's meant to analyze reference sequence, FASTA sequences. It's meant to give you rapid access to random parts in your FASTA sequence. It's meant to give you, to help you understand alignment formats. It's meant to give you quick access to random parts in an alignment format, to random parts of your alignment file. Yeah, we haven't done an alignment. We have a reference genome that we've just produced. Yeah, but in other tutorials, we're going to teach you how to create an alignment. So right now, the command we're going to be interested in is fadex. Fadex will generate an index and will gather basic information about the sequences that you've got. Why? Because since we want to grant it rapid access, yeah, it needs to know where each sequence starts, where each sequence ends, what's the total length of each sequence, that kind of basic output. So if we do this, SUM tools fadex, oh, yeah, we have to give it a file, right? Contigs.fasta. There you go. Apparently nothing happened, but if you double L, you'll see that there is a new file called contigs.fasta.fa, which was created 1 hour after everything else was created. And now, if you cut it, oh, yeah, let's just cut it. Yeah, if you show the contents, you'll see that it has 2 lines, 1 per sequence. We're interested only in the first 2 columns of that file. The first column is the name of the sequence, and the second column is the total length of the sequence. It doesn't matter, to SUM tools, it doesn't matter whether the naming
has or doesn't have information about it in the name itself. It will actually read the whole FASTA file and will get you what's the length. It will also tell you what's the byte offset, the length of each of the lines, and the number of total lines that are part of this. Yeah, but don't worry about it. That's these three last numbers there. Yeah, what we need to know is that node 1 has a length of 111,329. Yeah, you can do this with any assembler output you have. Any assembly output, you will see this. So have you already been able to execute the SumTools FadeX command? Did you all created the contigs.fasta.fai? Yeah, and you were all able to see it? Yes. Yes, all right? So it's telling us the actual sequence for this. Yeah, the actual sequence for this sequence, the actual AGTC sequence, it starts on byte 36. The second one is like, how many bytes per line should I use? Should I extract? It says I should extract 60 bytes per line. And how many bytes are there in every line? There are 61. Why? Because at the end of every line, there's a new line. There's a enter. The enter is also a byte. It's also recorded. Yeah, that's why it's counting it as an extra byte. Yeah, but yeah, talking about these bytes, things may be a bit confusing. That's why I didn't touch it. But if you want to know, that's basically what it's saying. Yeah, and in the case of the second one, it is telling you this sequence here, it has this length, and it starts from the start of the document. It starts on byte 113,256. Why? Because that's after the first sequence, after the second name. Only then it starts the new sequence, the new AGCT sequence. That corresponds to that name, right? And the characteristics are the same. It has 60 bytes per line are going to be sequence, and the last one is going to be just a return. Yes? Any questions? Basically, 61 is the total length of each line. It's just that each line consists of 60 characters, ACTG, yeah, plus a return, an enter, a new line. You'll see if you were to do something like this, head, just take a look at the first thing. You'll see that for every sequence you have a name, then you have a sequence of 60 base pairs, and then an enter, and then the second line, third line, etc. Each of these lines have 60 base pairs. But the length of the line is 60 plus 1 that you cannot see. It's the new line. It's the enter. Yeah, it needs to be stored anyway. So that's why the length of the line is 61, but the length of the sequence is only 60. The third one is, it's called the byte offset. Basically, it tells me, it's called the byte offset. Basically, it's telling me where within the document the sequence actually starts. Basically, when you see a 36, it means that the first 35 bytes of the document don't have any actual sequence information. It's just the name. The name and probably the new line that doesn't appear here. And this one, the first T, it starts on the byte number 36 of the document. Because that's how documents are read internally by the computer. It's byte by byte. Does it make sense? Cool. So let's say we want to just query a random part of this assembly. Yep. Let's say we want to see only the last 100 base pairs of this node. That's where we use, again, the same command, SumTools FadeX, but this time we give it allocation. It's better if we copy it this time, otherwise we're going to have a lot of trouble. Yeah, we put the name of the of the sequence that we want to target, that we want to visualize. And then we say the coordinates. So from where to where do I want to extract? Let's say I want to extract, I don't know, from position 1234 to position 2345. There we go. Yeah? So basically what we're saying is, first, we're telling, I want to access this sequence. Then we use a period, because we want to tell it from where to where. We want to see it from positions 1234, and I want it to finish at position 2345. So it's a little over 1,100 base pairs that we're trying to extract from this sequence. All right? And where is that sequence? It's not an enter, it's a space. Where is that sequence? That sequence should be in contigs.fasta. Now, the fact that we've already created this FASTA index helps me quickly find any spot in the genome that I want. I can have any reason to want to access that part of the genome. Yeah, but this lets me quickly access that. If I press enter, have you guys all finished writing here? Yeah? If I press enter at this point, please take a look at what I'm saying with this command. I'm saying use some tools, the command fadex, to extract this sequence, node 1 length, this, from positions 1234 until the position 2345 that is present in contigs.fasta. That's the minimum information I need to give. If I don't say where should this command find the sequence, it will throw an error. If I don't say it has to be in contigs.fasta. If I throw in the wrong the wrong FASTA file, yeah, for example, a FASTA file from another assembly that doesn't contain this exact name of sequence, you'll get an error. Yeah, so you want to access that, you just press enter. Oh, no such file name or directory. Why? Failed to open over. Oh, sorry, my bad. The order is wrong. I should have put here. It should have been. Sorry. Yeah, you have to put the config name, the file name before the actual location. My bad. And there you go. It has extracted that part of the genome. If you try to extract the same region by any other means, yeah, there are a few other programs that may be able to do something similar, but SAMTools is the fastest one. You can randomly access any part of your genome or your FASTA file quickly without many problems. Now, we're going to leave, the next part we're going to leave it for now. We're going to stop this today. Yep. The next step is usually validation. But for validation, we need to run quite a few more things that may take us half a day more to finish, right? But I'll quickly talk about what validation means and what are the three main types of validations that we need to look into. All right? So this is where we are right now. We've finished an assembly, right? So there are three main ways to assess the correctness of an assembly. Because an assembler could actually, the first thing it could do is just concatenate all your FASTQ files, and instead of assembling them, like finding alignments, etc., yeah, the only thing you do is just you put one sequence after the other, and you get one single contig in the end, and it tells you, yeah, it finished. Yeah, but that's not actually biologically real. That's just an artifact of the assembly, or of the assembler. A bad algorithm will generate a bad assembly. Yeah, and even though some of the metrics will be good, will look good, that doesn't mean that the assembly is correct. So there are three main things that we need to look into before deciding whether an assembly is correct or not. The first one are called the contiguity metrics. Contiguity metrics are meant to tell us how well the repeats have been resolved in your genome. For example, let's talk about first total size. If you know that your genome was 150, or was expected to be 150,000 base pairs, and your assembly it's 700,000 base pairs, yeah, do you think that you reach what you were expecting, the total size? If you have an assembly that is 700,000 base pairs, but you expected just 150,000, how many times over have you repeated have you assembled the genome? Where does all the additional data in your assembly come from? If your genome was originally 150,000. If you take a look at your files, you'll see that your assembly should be around the 130,000 mark. That's pretty good. You're expecting to get a genome of 150, and you got an assembly of 130. That means that you've assembled nearly 90% of your genome. Yet there's a part that is always going to be missing. Those are the gaps, the unknown regions. Why do you have those gaps? Because these regions have, I don't know, maybe they are difficult to capture, maybe they are difficult to amplify in the library preparation protocol. Maybe they are too repetitive, so that even though they are captured during sequencing, they cannot be properly assembled. Yeah. So all these are factors that need to be taken into account when deciding whether you have a good assembly or not. In this case, if you have assembled roughly between 80 and 100% of your expected genome size, it should be okay. It's telling you that, yeah, the assembly looks good so far. Now, N50 and NG50. No, let's skip that. Let's go to number of contigs. The number of contigs is going to tell you how fractionated your assembly is. That is, if I have a genome, an assembly of 130,000, and I have it in 1,000 contigs, that means that each contig is going to be very small, on average. It's going to be very small. Yep, that's not good. Because usually, if the expected size of a gene is longer than the average size of your contig, that means that most of your genes are going to be split across different contigs. If you get that, then you will not be able to mine genes or to find complete genes in your assembly. And that's one of the main reasons why we do a genome assembly, to find genes. If a single contig doesn't contain at least one single gene, then your assembly is not going to be very useful. Next one, that's the next one. It's average contig size. Very easy. You just divide the total size of your assembly by the number of contigs that you have. And you have an average contig size. If that average contig size is not above the expected gene length, then you have an issue there. The bigger this average size of contigs is, you can pretty much assume that it's a better assembly, a more contiguous assembly. Because you were able to find more contiguous regions that could be assembled together into a single contig. And that's why you have bigger contigs. Yeah, that's basically what it means. Now, N50 and NG50. Basically, this is kind of a sorting analysis. You sort all of your contigs by size. from the biggest one to the smallest one. All right? Just follow me on this one. You sort the contigs of your assembly, you sort them by size, from the longest one to the smallest one. And you start summing up how many bases have been assembled in the first contig, then in the first 2, in the first 3, in the first 4, in the first 5, 6, 7, etc. You keep summing up, right? You generate an accumulated assembly size from the biggest to the smallest contig. And then you say, all right, when in all this did I reach half of the assembly size? What was the last contig that I added to this cumulative length where I reached or surpassed half of my assembly size? In the case of our example, the assembly size it's probably around 130,000. Half of it should be 65,000. Yep. If we start summing up every contig from the biggest to the smallest, in our case we only have 2, yeah. In what contig did we surpass the 65,000 mark? In the first one. Yeah. So the size of the first contig, that's my N50. That size, the size of the contig where I reached or passed half of the assembly, that's my N50. Similarly, for NG, yeah, we're not using now, as a total, we're not using the assembly size, but we're using the expected genome size. So the expected genome size for us was 150,000. That's what we were expecting to get. That's not what we got, it's what we expected to get. So at what point, when we start adding up the sizes from the biggest contig to the smallest contig, did we reach half of that expected length? Half of it would be $75,000 base pairs, dollars. That's our salary. So half of that would be 75,000 base pairs. Yeah. So in what contig did you reach or passed the 75,000 base pairs in your examples? In the first one. The first one is over 100,000. So that means, again, your NG50, it's the first one, or 111,000 something, right? So these are contiguity metrics. These are just meant to kind of more or less describe how big your contigs are, how this length is distributed among the biggest contigs compared to the smallest contigs, etc. You can use different set of metrics to do this, but these are usually the main ones that are reported. Now, just because we have big contigs doesn't mean we have a correct assembly. As I put in the previous example, and I'll do a very quick example here. Yep. Just a very quick example. If we have this one, and then this one, all right, we can see that there is equivalence here, 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 and here. There's an alignment there, right? So we could say the assembly could have dropped the following result, which would be just so the assembler could have said, yes, that's my assembly, because I found the overlapping regions, I got the consensus sequence, and yeah, that is my contig. But another assembler could have equally, just because it was a bad algorithm, could have done this. Just concatenated both of them. It is not really assembly, right? You just concatenated two strings, yeah? But if you just look at the contiguity metrics, you'll see that what's the average size of each of the assemblies? The first one got me an average size of, I don't know, 10 base pairs probably. The second one had an average size of 15 base pairs. The second one is bigger, no? It looks more contiguous. That doesn't mean it's right. This is actually completely wrong. And that happens all the time. That happens all the time with different assemblers. Every assembly has some errors. They are not easy to deal with because they are just inherent parts of each algorithm. And each assembler has its own way of handling them internally, trying to find out which are misassemblies and try to just remove them from the whole assembly. But just because you have good contiguity metrics, which is what is most commonly reported in every genome assembly paper, that doesn't mean that you have a good assembly. That's why we go and we rely on other metrics. These are the coherence metrics. The coherence metrics basically let us find out how the reads we use for an assembly actually are present and are distributed across our reference. And for that we use a technique called mapping. Mapping basically, it's an algorithm that makes a very fast alignment between the short reads that we use as input, the FASTQ files, and the reference assembly and the reference genome we just tested. It uses an algorithm called Burrow-Wheeler's transform, which basically it's a fast algorithm to look substrings within a longer string. That would be the most simple explanation of it. It is merely a computational or an IT algorithm. It was just taken by the bioinformaticians in order to do this kind of analysis faster. But essentially it just indexes a reference genome, and then uses this Burrow-Wheeler transform to actually do the search of these sequences, which are the FASTQ sequences that we have, as fast as possible. You can very easily align 10 million reads to a human genome in less than 1 hour, which you wouldn't be able to do using any other algorithm. Other algorithms you do know for aligning things, would be BLAST, for example. But have you ever tried to run a BLAST of 10,000 sequences online? How long does it take? It takes forever. There is no way you can scale that, scale to this level of millions of reads towards one big genome in such a short time using an algorithm like BLAST. That's why they had to come up with new algorithm or use recycle algorithm that already existed, in the case Burrow-Wheeler transform, in order to make the alignment faster. So once we align the reads back to the genome, we start checking how well do these reads align to my reference genome. And what should we expect? First thing, if the assembly is good, if the assembly is good, I expect that most of the reads that I used as input should be found in the assembly, because those reads are the raw material that the assembler used to build the assembly. So if you have very few reads of your input reads mapping to your assembly, then there's something wrong with your assembly. That's what we call mapping efficiency. Mapping efficiency is basically the percentage of reads from the total reads. Let's say you had 100 reads. If from 100 reads, only 30 map, that means you have a 30% mapping efficiency. How is it possible that only 30% of your reads map to a genome you just assembled with those same reads? You should expect something around the 90% or the 95% mapping efficiency. If you have something lower than that, there may be other explanations. Presence of repeats, or contaminants that you removed before assembling, etc. But at least from those reads that you use for the assembly, yeah, you should find most of them in your that have been mapped to the reference genome. At least. Most of them should be there. If you have less than 60% of your reads mapped to your reference genome or to the assembly, yeah, something went horribly wrong. Next one, properly paired mapping efficiency. What does properly paired means? This means that you know that the reads are paired n, right? And by paired n, we mean that each extreme, the forward and the reverse, should be facing to each other. And not only should they be facing each other, they also have like an average insert size. That is, there is an average distance between the forward and the reverse. Some of them can appear tight, some of them can be a little farther apart, but most of them should fall within this range, right? They should have a certain distance. Now, what happens if most of the reads that you map to your reference genome, yeah, they are facing outwards when you look at them? What happens? Is that a good, the sign of something good happening? If you were expecting all the reads to be looking inwards, why would you find reads looking outwards? That indicates two problems. Either you have a poor assembly, or the mapping didn't work out as fine, right? It didn't work correctly. So the first thing I would do, I would double check all the parameters of my mapping algorithm, and if they all look good, let's say, okay, then something is wrong with the assembly. This is also a coherence assembly. Finally, the third most important coherence assembly metric is the total coverage. We already assessed that during the k-mer stage. We know how often each k-mer should be present on average on the reference genome, yeah, on the expected size genome. In most cases, it was around 666. In my cases, it was around 37 times. Yeah, there is a little bit of error, standard deviation around that number, but more often than not, most contexts should have that coverage. So if I map the reads back to my reference genome, I would expect that for every position in my reference genome, I should have more or less that coverage, or more or less that depth. So if I was expecting 30x coverage, or 30x total depth, that means that for every position in my genome, I should have around 30 reads aligned to the same position. If we don't have that, if we have regions of the genome that have much lower coverage than that, or much lower depth than that, then that's indication that there is something wrong with that position of the genome. For some reason, that position of the genome doesn't have enough reads represented. It is possible, and this is just a conjecture, it is possible that maybe this region is underrepresented in the library construction method we use. Yes, it is possible. Yeah, but it's unlikely. It is unlikely. There are other methods, external methods to test that. If you find a spot that have either way too much coverage or way too little coverage for what you were expecting, then you can design primers around it and try to do a PCR. If you get a result, good, and you sequence the result using Sanger, then good, it looks good, then there was something wrong with the library in that position. There is something particular at that position of the genome that doesn't allow it to be sequenced easily. But if you don't get any PCR results, the most likely reason is because that region doesn't exist. It is an artifact of the assembly process. The algorithm, for some reason, misassembled this part, and it's just making this up. It happens. Trust me, it does happen more often than one wishes. Finally, completeness. How complete your genome is. And here you'll use a combination of different metrics, but mostly use one tool called Busco. Busco is benchmark of universal single copy orthologs. This is because we want to measure how complete our gene space is. And what do we mean by gene space? Is that from the reference genome, we know that some parts are coding regions, like genes, and some parts are not. We can mostly measure the parts that are gene-rich, right? So we say, okay, what are the genes that appear more commonly between all the species of this particular clade? That is a Busco dataset. A Busco dataset has been compiled in such a way that it just selects the most common, the most universal set of genes in a specific clade. It could be either mammalian, it could be bacteria, it could be prokaryotes, it could be archaea. Well, but it selects the most universal set of genes that appeared only once per genome. They've done a very extensive database search to know and select which genes appear in nearly every single individual of that clade, and not only that, but appears only once in each genome. So that means that there is an evolutionary constraint that this gene is usually never duplicated, and secondly, there is the evolutionary constraint that if it remains in several individuals of the same clade, it is so important that it cannot change very easily. It cannot disappear. It has to be evolutionarily important. That's what a Busco assessment is. It has that training set, that set of genes that another group has already searched. It's a public database. If you Google Busco genome completeness or Busco annotation, you will see that you can download the database that you want because somebody has already done this exercise. Yeah? And depending on the genome you're working with, you can download a database that lets you estimate what is the gene completeness. If you have more of these genes that are considered essential, there are single copy orthologs that are considered essential for your genome, and that most other individuals in your clade, in your evolutionary clade, yeah, also have, then you say, okay, from the gene space, it looks complete. If you have fewer, then it means that you're missing that spot. It doesn't necessarily mean that your genome assembly is bad, but it does mean that it doesn't contain as much genetic information as you want. And more importantly, it doesn't contain the gene coding regions that you want. Because that's the main idea of behind assembling a genome, to get the coding regions. So RNA Sig mapping, and this is just an if you have RNA Sig experiments, that is transcript and full transcriptome experiments from the same individual you have or from a very close relative, yeah, you can try to map this into your reference genome. The higher the mapping efficiency, that means more of these RNA sequences are present in your reference genome. And that means that the gene space of your reference genome is more complete. Similarly, with transcript alignments, if there are already cDNAs or full-length cDNAs that have been constructed for your reference genome or for your species, you can use them, map them to your reference genome, and see how many of those full-length transcripts are found in your assembly. Please take a look at all these are using external data. They are not based solely on the genome assembly data itself. We're using either a public database of single copy orthologous regions, genes. We're using RNA Sig data if available. We're using full-length transcript data if it's available. And you can think of anything else. If you have a SNP tags, somebody has already found SNPs in your species, and they have uploaded a SNP tags, that is small sequences where the SNPs are present, into a public database, you can download that, map it, and see, oh yes, I have this many SNP tags in my database. The higher the number, the better the assembly is, or the more complete it is. If somebody has produced a back-end library, and they have sequenced back-end sequences for each of the backs, you can also download the data, map it, and see how many of the back-end libraries, back-end sequences, you can find in your assembly. Also, just like before in Coherence, each back library will have an average insert size. So and also both the forward and the reverse reads of a backend library should be looking inwards. Yeah, so if you find those, if you map those reads or those sequences to your genome, they should be facing inwards and also should have an average distance as is expected from the best library. The same for PostMit libraries. The same with ESTs, express sequence tags. All this external information is used to assess how complete your genome is. And in every single case, the higher the percentage, the better the assembly, the more complete your assembly. And Busco, yeah? And you don't choose it. You don't choose it. Yeah, basically each data set, yeah, because they have already generated databases, yeah, for each of the biggest clades. If any of you want to open the page, you will see that you have like 7 or 8 of the biggest clades. You have, for example, you have the mammalian database, which uses all available mammalian genomes. Yeah, it searches for all the genes that have been annotated there. It looks orthologous sequence between them. Yeah, so they found orthologous between all the genomes, and then they search which of those genomes were present on average only once in every genome. And that's the data set that they selected as a gold standard for mammalian genomes. So the next time you sequence a mammalian genome, yeah, you'll go to that data set, yeah, you'll check if those genes that are in the data set are present in your reference, in your assembly, yeah, and the more of those genes are present, it is an indication that your assembly is more complete in the gene space. Oh yeah, though, the idea that there will be a difference in the nucleotides, there's definitely going to be a difference in the nucleotides. The main thing about the way we look for it is we look for it using an algorithm kind of like BLAST. Yeah, it allows for certain differences between the original database that you're looking for and the actual sequence that you get. So basically, the way these essential genes are found, yeah, is flexible enough to find a sequence divergence that is high, but the function remains. That's the way this assessment is done. All right, so we're going to leave it there. For now, if you have any further questions, please don't hesitate to contact any of us. I know there's a part missing, the genome assembly, but we don't have the time to go through that at this point, unfortunately. It's going to take way too long to actually do a genome assembly. That's fine. So I'll just leave David take after this. If you have any questions, please don't hesitate to ask it.
Can you all hear me? Yes? Okay, very good. Yeah, so while you copy this, let me say a few words first. First of all, I asked you to download USearch. Who managed to install USearch on their computers? No one. Only the Mac guy. That's good. Okay. So apparently there is a new version of it called vSearch that I'm not aware of, but it works the same. So if some of you have problems installing that on Windows, maybe you can try vSearch instead of USearch. If that's not going to work, then we need to somehow use it on our Linux distribution, the Biolinux that we have. Okay, so while you're copying, let me say a few words not regarding USearch. So this course is very intense. We only have 2 weeks time here, and we're trying to open the door for you guys to see what kind of tools are available in bioinformatics. The field has grown so much in the past 20 years that you have subcategories for bioinformaticians right now. So I'm basically not what Juan is. I'm a network specialist who's working mainly on metabolism. And that is another field, probably the most recent field that has grown in bioinformatics. And I know it gets very frustrating sometimes that you're trying to do stuff and you're not used to it. And Linux is not very user-friendly. It's a very hard tool to get by to. It's hard to get accustomed to how it works, the different commands that you have. There are thousands of different commands, and trust me, I don't know even 1% of them. So even in here, I learned a few new things that I didn't learn before, that I didn't know before. So you can always extend your knowledge. But one thing that I would like to tell you is you need to get your basics right. And the basics, I would say right now, is to learn Linux, and the other one is to learn R very well. When I started programming about 19 years ago, it wasn't that easy as it is today. Nowadays, you have the opportunity to look up. Basically, you can ask every question that you have, and someone else would probably has worked on it already. So you have Stack Overflow, or you can just Google into it. When I started programming, there was no such thing. I remember the first time we went into the library, there was this old librarian, and he told us, there's this new search engine. Did you hear about it? It's called Google. So that was 19 years ago. We all used Yahoo. No one else used Google at this point. And it has changed so much. Now Google is the primary search engine for everything that you will use. There are other ones, but Google is so strong, and it's also a very, very good algorithm. Basically, always the first 2-3 results will display what you're looking for. That being said, so there would be people who tell you, forget R, do something else. When I was starting to program, Java was the big new thing. And everyone wanted to use Java. And Java is a very nice tool, and it has also manifested itself in the world of programming, but it's a very slow tool. So they told us back then, forget C++. No one will use C++ in 20 years. They told us, forget Linux. Everything will be done in Windows. So the truth of the matter is, C++ is back, and Java has lost a little bit of its, how would you say, presence in this world. So, and Linux is going up again, because it is such a useful tool, and it's actually, when you get by it, it is very, very easy to use, and it's so convenient to use. Like if you have to go with your mouse, if let's say you have to do some things 1,000 times, and you have to do it with your mouse, you will get nuts after 2 minutes. Trust me, I had to do it, and it's not very practical. So Linux lets you do stuff that you do in an hour with a normal computer, probably in a second, or even less. So it's a very, very useful tool, and I would recommend that you get by it, you try to learn it, and you try to master it. And it doesn't mean that you have to know every command that exists. It has to mean, what it means is that you know how to use it. So if there's a new command coming that you never used before, then you know how to use this command, where to look for help for, and what these different parameters that you have in these commands tell you, and how they help you. So that being said, I would like to show you, before we start going into the lectures themselves, now that I was walking around, I saw people doing some stuff, and they still didn't know what it was. Can you all see that, or is that too small? I think it's too small, no? No? Doesn't work? Okay. Is that better? Can you see that? I will. Wait. Bigger? Like this? You can see that? Okay. Juan was already talking about this, is the double dot and the dot. But I will repeat it because I think it's not so clear yet. So if I want to list the files that I have in my current folder that I'm working on, I can use the command ls, right? It would show me which commands are there, which documents are available. If I want to look at it in a way that it gives me a bit more details, I can use ls-l. So then I will know the size of the documents that I have, and it will tell me whether it's an executable, whether I have read-write, or I have write-write, and so on and so forth. So what we usually do is, instead of using ls minus l, there's a quicker command, but it only works on Linux, so I cannot show you this. And it will give you the same thing, but it's just ll. So if you do ll, it will give you ls minus l, basically. Another way, what you can do is add h, which means human. So here, instead of telling me 238 what, 770 what, it will tell me it is 777 bytes. It is 576 bytes. If there were files that are in megabytes or in gigabytes, it would put an M, it would put a G. So it's more readable for you. Instead of giving you a very long line, you will understand better. And why is this so important? If you are working on a server and you want to execute a bioinformatics pipeline, you need to know the sizes of your files, because you can mess up the server very, very easily if you don't take into account the size of your documents, the RAM that you want to use, the numbers of CPUs that you want to use. Speaking of which, if you want to use, doesn't work on mine, huh? Probably only works on Linux. When you want to know how many CPUs you have available, you put ls CPU. And there are other commands that tell you exactly what the size of RAM of the computer that you're working on. Because if you're working on a server, you will not get a fancy graphical user interface. You will have to work with the shell. And the shell will only show you these information. Okay, so I was talking about the dot and the double dot. So what I can do is, if I want to say ls minus lh, in the current folder where I am, I just put a dot. Okay? If I want to look at the directory that is above where my folder is inside, I put 2 dots. And now it will show me all the files, among others, also the folder that I'm working in the directory above. If I want to go 2 directories above, I put a backslash, another double dot, and now it will show me exactly the directory that is 2 directories above. And here we'll see it shows me 1.3 megabytes, 384 Ks, 15 megabytes. So this is what the minus h will tell you when you use ls. This is important because it also works for R. If you want to see in R what your current directory is, then you can also use dot, or you can use double dot to go one directory above. You know, that only works for Linux, it also works for R. Tilde, something that you haven't before. Okay? If I use the cd command, you already know that I can change into different directories. Here I don't have a directory, but if I want to get a directory up, I can just say cd double dot, and now I will be in the directory above. If I just use cd, and I look where I am, it will go to my home directory. If I just use cd, it is like a reference that I always will go to my home directory. Okay, this home directory is stored in the tilde. This thing. So if you are programming, or you're using one of the Linux commands, and you want to specify a certain directory, and you know it's closer to my home directory, you can use the tilde. It will go then to users David, and then it will show me what there is. Instead of using the entire path, you can just use the tilde. But that always represents your home directory. Okay? Clear on that? So everyone has managed to copy the files? Okay? So in these 2 folders, there are the lectures that I was planning to go over, but I don't think I will have the time to do all of them. But when I prepared those lectures, I wasn't aware of the level that you have here. So I assumed the worst. I was positively surprised. Most of you have a very high level, and most of you are already established researchers. So these lectures are very basic. It's a very basic introduction about how to use graph search, how to do multiple sequence alignments, and how to do phylogenetic trees. unfortunately, I don't think that we will have the time to go over all of them. But you can have them, you can look at them, and this is also a reference point when you have questions. That, for instance, who ran a blast already in their life as a researcher? Show of hands. Blast. Blast. Yeah? Who knows what they're doing when they're running blast? Yeah? Okay, that's what I was expecting. So when you run a blast research and when you go to NCBI, you will have those different options. You can run a local blast, you can run a global blast, global search alignment, you can run it with a PHI database, you can run it with a PSI database, you can run it with a Blossom 62 database, you can run it with a Blossom 50, you can run it with PEM 10, PEM 20, PEM 30, up to PEM 250. What does it all mean? Anyone? Idea? So, and this is what you have in those lectures. These lectures will tell you what these different options that there exist when you run a blast actually mean. So that when you run a blast, you know what you're doing and why you're doing it. Because sometimes if you, let's say, if you compare two sequences of proteins, amino acid sequences, and they're actually from the same kingdom or from the same species, you want to use a Blossom 62 database, which is 62% of identity. If they are very far apart, you want to reduce the Blossom size. Maybe you want to go to Blossom 31 or something like that. Means you are presuming that there's only a 31% identity between the two proteins that they're using. So this is what you will find. This is some of the aspects that you will find in the lectures that are provided in the lecture file. But we don't have time to go over this, thanks to Juan. No, just kidding. And I was thinking, since you already have experience, we would do something that would be more useful to you. When I talked about the basics and the fundamentals that you need to learn, I think that we need to cover 2 or 3 more aspects in R, which will make your life easier if you know how to use them. Okay, so also what I wanted to show you, if you see here, I basically always have open a few tabs when I use my shell. Okay, you can do the same. And you should do the same. Why is that? So let's say I have here in my bash, I'm looking at my files, I want to know what files I have here, but then I want to do some quick R operation. So R, as Juan already showed you, that if you enter R, you have the R console open, and you can do, and you can run R. And then I can use my R command, and then I go back to my other shell and say, aha, I know what I did there in R, so, or I want to see what route I've been using, so I can just take the route from here, I can copy it, and use it here. Let's say I want to change my working directory. And I want to check where I am now. And just by copying the other shell where I was in, I can open another shell. Let's say I want to make sure that I know what I'm working on in one directory, but I'm still in the other directory, but I don't know what the route is. So I can combine all these little tricks, but very common, but it makes your life easier. Let's say I want to, another trick that you could use, I want to use the manual on CD, okay? Ooh, gives me so much information. What can I do? So I can just write, for instance, the entire manual into a log file. So I say man cd.log. So the arrow, the greater than arrow, is an assignment to a new file. So right now, I don't have this file cd.log. Okay, I only have these files. Okay, but when I do the greater than arrow, I can cd.log Okay, it will create a new file with the entire content of manual of CD into the CD log file. If I look at it now, you see CD log, right? And now I can go into my text file, the text editor. Where was I? Home, right? Yeah. I wasn't. You see it? No. Should we use this? So it should be here. There it is. Okay, so now it displays me all the information. Horrible, right? Yeah, okay, sorry. So but now I can go back into my shell if I don't know what to do, and then I can look, here are the different options. You can do this, you can do that, you can do this, and you can do that. Okay? So with a simple assignment, yeah. It's very, okay. Yeah, I did have to use a different interpreter when it's right. But anyways, what I wanted to show you, with the greater than command, you can easily assign the content that you have here into a text file, for instance. If I want to double this one, will add to the end of your file. Okay, if I do this again, it will overwrite my file with something else. It would still be the same file, but the content of my former command is gone. So what I want to do, let's say I want to see both. First I create this one, and then at the end, with a double greater than, it will add to the end of my file that I used before. That's one before when Juan used grep. It was very important that you use actually the quotation marks to put the greater than sign in, because otherwise it would have just put grep into a file that you specified later on. Happened to me several times, and I lost all the data that I had, so be careful with that. Okay, now, if I want to, you saw that you can start R by just typing R, but there's also the option that you want to execute a file from your shell script. Let's say you have a file of R commands and you want to execute it from your shell script, you can use the command R script. You can pass here arguments to your R file. Later on we'll see exactly how to do this. But again, if you're working on a server, this is how you will need to execute your R script. You will not have the option to run RStudio on your server. Of the file, of course, yeah. Yeah, script. Something like that. Doesn't matter. You will see later on how to do this exactly, and then you will get an idea. At the same time, if I open RStudio, I'll make it bigger. I can use the command in R called system. And this allows me to execute shell commands, or in your case, maybe window commands. Intern. So let me execute this first. If I want to clear the console, I can just do Control L. Okay? Okay, so I'm using, if you're looking here, can you all see this? Where is it? Where's the laser? Okay, so this is my function system. I'm telling it use the ls command from Linux and give it an output intern equals true that fits my R. If I put F, it will give me an output like Unix will let me, it will give me. So, and then my output is, I have those different files in my current directory where I'm looking at. So let's say you have a very long R script and you're thinking to yourself, instead of doing this in R, I would like to do this in Linux because I know there's a Linux command that can do it much, much faster than I can now try to program something in R. So let me just use the Linux command, and then I will get back my data, and then we'll continue working in R with it. So there's a chimera between R and Linux, where you can use Linux to execute R commands, and you can use R to execute Linux commands. So this is something very useful, and I would recommend that you do this if you get into this. Okay. Now, so we haven't discussed conditional statements yet. And when I mean conditional statements, I mean if statements. Anyone familiar with if statements? You can actually use it also in Excel or in any other spreadsheet program. And it's a very, very simple comparison between two conditions. Let's say I want to compare two numbers. I start with using if. Now you can use RStudio. Everyone open RStudio? Yeah? If 5 greater 3. So in between parentheses, I will put a condition. And the if statement tells me if this is true, whatever is in here, then do something else. So is 5 greater than 3? Yes or no? So I want to print something to the screen. Obviously, you idiot. Okay, so if 5 is greater than 3, it will print this message. Okay, let's see if it works. Obviously, you idiot. Yeah. Let's say if 5 is smaller than 3, what will happen now? I just changed from greater than to smaller than. What will happen now? Anyone? A guess? Will I get the same message again? Obviously, you idiot. Who says yes? Who says no? Who's undecided? Let's try. So, nothing happened, right? It just shows me the script that I executed, but there's no decline, obviously. It is because 5 is greater than 3, so how can it be true? So what happens here, you just want to look at what if the if statement is, if this statement that is within the parentheses is true, do the following, which is in the scope that I'm defining here. The scope is you open with curly parentheses and you close with curly parentheses. So, let's do this again. If, tell me something. I can compare if A is equals to greater A. What do you think will happen? So the double assignment of two equal signs doesn't mean assign this to that, is compare. Is this equals to that? Who says that it will be true? Raise your hand. Yeah? Who says it will be false? Who's undecided? The majority. Okay. And nothing happened. Because a lowercase a is not the same as a uppercase a, right? So the computer says this statement is not true. So I want to tell the computer, don't just do something if it's true, do also something if it's not true. So I continue my statement with else. And I do the same. So if a is equals to a, lowercase a is equals to uppercase a, print the message true. If it's not true, else, print false. So we already know what the answer will be, right? Right here, false. Okay, so I can easily check whether if the condition that I put in parentheses after my if statement would be true or would be false. And this is the idea of an if statement. So you have the option of doing it with a greater than, a smaller than, or equals to compare to have a comparative condition. And this is exactly what an if statement is. Let's make it a bit more complicated, because obviously if I put something in like this, then it doesn't serve a purpose. Let's say I create two variables. Variable x is equal to 10. Variable y is equal to 12. Okay? So I have my two variables. If I look at it, x is 10 and y is 12. I ask the computer, compare x to y. Are they equal? No. What will be the output of this? What will be the output of this statement? Who says false? Very good. Because x is 10 and y is 12. So instead of doing, you're putting in two variables that you assigned values before, and the computer looks at those values that are in those two variables, makes the comparison, and then tells me whether my assignment, my comparative assignment, is true or false by doing this. Okay? Now, let's say, okay, so I told you before, I can execute an R script right from Linux shell. Okay? And I can pass arguments from my shell to my R. How does R know what the arguments, the parameters were that I passed? For that you need this the function command args. Trailing only make it true, because otherwise you will get other arguments that you will not want to use. So then I get the arguments that I have for my Linux into my R. And now I have the variables called args for arguments. And now I can say x is equals my first argument, and y is equals my second argument. And I will show you now what would happen if we execute this file through Linux. You can actually do it. That's a new feature of RStudio, is that you have a terminal right built into it. Down here you have your console and you have your terminal. Okay? Everyone can copy this code. Once you've finished copying the code, save this as an R file. Okay. So I will save it as demo.r. Okay. Now you see that the name of the file has actually changed from untitled to demo.r. Everyone ready? Yeah? Okay. I'm going back to my shell. And you can just do it by going inside the terminal shell. You don't have to go actually into the shell itself. You have a shell. No, you don't. He does have a shell.
Remember guys, after the if command and the condition, you have to open and close brackets.
I will put it back. So. If I find it.
Look, after the if parentheses x equals y, close parentheses, you have an open close bracket. That is important because that's where your actual commands are going to be placed.
Right.
If this is true, then when L outside of the curly bracket, open curly bracket again, is false.
Like why are those curly brackets so important? Obviously now I'm just showing you how to use an if and else statement, but sometimes you want to put a bunch of lines into one single scope of an if or of an else. If this is true, then do this, do that, do this, do this, do this, do this, and this and that. So the computer needs to know where I'm starting my if and where I'm ending my if. And if this is false, then do this and do this and do that and do that and do that and do that. So it's very important to always put your curly brackets. You can omit putting the curly brackets, but what would happen then is it would only execute the line right after the if statement. All the other lines that are meant to be in your if scope will not be executed, only the first line. So it's a good, like I've seen many programmers that just put without curly brackets, but it's a good exercise to always put your curly brackets. Otherwise, let's say you're programming something and you want to change it. Then you will have to put the curly brackets again because you're adding a few statements into your if. Everyone managed to copy this code? Did you manage to save the code? Yeah? Shall we execute the code? Okay, so let's go back to our shell. Make sure you're on the right one. You don't have to, but I'm just showing you to make it so that you know how to, in case you have to. What's that? You have the power Windows something? Where was I? So I'm going into my directory, which I cannot find right now, day 6. Okay, more R. Why not? Excuse me. Let's see, I have demo R here. So if I type R script demo.r, okay, and can everyone see this? So I want to invoke an R script. Which one do I want to invoke? The demo.r. Now I want to pass a few arguments. So let's say I want to pass 5 and 5. What will be my output? Anyone want to share a guess? What were we trying to do in our R script? We were comparing x to y. If x is equals y, it is true. If x is not equals y, it is false. So I'm putting 5 and 5. Is 5 equal to 5? Yes? True. Okay? Let's say I'm changing to 8. It's false. But they should have the terminal in our studio itself. But no, I mean here, no? No, here the terminal. Here terminal. We need to save the file. See, when I created this file now, you see that I called it demo.r. So this, if you don't save it, you cannot execute it. Acá tiene un terminal. ¿Viste? Allá. Se metieron. Sí. Acá. Ahorita cambia en tu carpeta donde estás acá. Y haz. Okay, let me demonstrate this. I can use the terminal right from RStudio. Okay, so instead of going to my shell, I can do it actually in RStudio. So let me see where I am. Yeah, okay. So I want to execute R script on my file demo.r, and I want to pass two variables, one 7, the other one 8. So you don't need to go into your shell. You can use it. You should also, you have the latest version of RStudio? So before you can actually execute the file from shell, you need to save the file first. As long as you only have it in the editor of RStudio, the computer doesn't know it exists. It exists on screen, but not in memory. So in order to execute it, you need to make a file called demo.r. I want to show you, you can make more than one statement in one if. So let's say if x is equals to true y, then I want to write to the user they are equal. Let's say if x is bigger than y. So I can extend my if statement by saying else if. Okay? If x is bigger than y, then I want to write, and I can use the paste function that you've seen with Reinhard already. Then I put x a comma, a statement is greater than comma y. So what will this function paste do? It will concatenate my variable that I have in x. Say, let's say I put x, I put a 5, it will say 5, and then it will write is greater than, and in y I put a 3, is greater than 3. So first I'm comparing whether x and y are equal. If it's equal, then write they are equal. If x is greater than y, then write to me x is greater than y. Or whatever is in x is greater than y. So there's only one last option available, which is they are not equal, x is not greater than y, so what must it be? Y is greater than x, right? So I'm putting at the end else. So if nothing else can happen, I'm copying the statement, print, and I'm just changing the variables. Y is greater than x. I'm saving it, because only by saving it, I'm updating it. Okay? Now I'm going into my shell. I'm here in my shell. Now when I say 7, 8, 7 will be assigned to x, 8 will be assigned to y. So what will be the outcome? What will be printed to my console, to my shell? The lowest one. Right. 8 is greater than 7. Right? Now if I change the variables, sorry, 8 and 7, what will be printed? Actually, the same message will be printed. 8 is greater than 7. Right? Why is that? Is the second one, but the same message. So it's confusing. So for me as a user, like I don't know what to do. I just want to know, is x greater, is what I enter, I don't know if it's x or if it's y. Whatever I enter into my console, I just want to have the bigger variable. Or if they're the same, just tell me they're the same. I don't care whether you say first x is greater than y or y is greater than x. I just want the result, right? This is what you do when you use a program. You use a program. You don't want to know how the program actually is doing what you're asking for. You just want the result. And this is programming. So it's a very, very simple program. But this is actually how programming works. You can make those statements much, much more complex. You can combine statements. You can put an AND. You can put an OR. Yeah, let's say I want to know whether x is equals to y, or, and this is my OR symbol, a vertical bar, x is greater than y, then write to me they are either equal or I have to put it in a paste 0, or, and I put it in the next line so you can see it better, x is greater than y. Okay, so save it. So what would happen now? So I'm saying now, is x equals to y? Or is x greater than y? What do you think will happen? Let's say I put x7 and y7. What would happen? Will I go inside this scope? Okay. It's a question. It's just a verbal question. Is it true when I ask you, are you a man or are you wearing glasses? Is both true? Right, both is true. And I ask you, are you a man or don't you wear glasses? Well, one is true, so the whole statement will be true. That's an or. Okay? It's not a pipeline here, it's a vertical bar. Vertical bar means or. Okay? Is either one true? Is either one true? Then do that. But now I have also here x greater than y. So I have the repetition of what is happening there. So will I have two statements? They are either equal or x is greater than y, and then again x is greater than y? Or will I just have one statement? So what will happen is, it will say, I'm looking here, this is true, I will ignore what it is, and I ignore the else. No, it's not necessary, this is how it works. So I can say, if this is true, one of those is true, ignore what is here and ignore what is there. If this is not true, look what is here. If this becomes true, ignore that. If this is not true, if that is not true, then do this. So it's like if or else. You cannot do both. Okay? Vertical bar or. Okay? Try it. Save it. So what I would save it. So again, I will execute 8 and 7. X is 8, y is 7. It's like they're either equal or 8 is greater than 7. Okay? Now I put 8 and 8. They're either equal or 8 is greater than 8. Obviously it doesn't make any sense, but this is the way I programmed it. And I have control about how I program it. So obviously, I can extend my if statement. I can make 3 ors, I can make 4 ors, I can make 5 ors, as many as I want, actually. Yeah, that was my next one. So, in an or statement is, if either one is true, the whole thing becomes true. Now we can do AND. But this doesn't make any sense. So AND will only be executed if both are true. Okay? So it cannot be, why can this not never happen? Now as it is, can both be true? Why? Is greater, exactly. So let's see. So if I put the same variables, it will tell me is greater than, because it went in here. It went into this statement. And here so let me change this. So you believe me what I'm saying. Something else. So, since this cannot be true, this is also not true. I put 8 and 8. It has to go into this one. It doesn't. I don't give it another option. Okay? Let me change the variables. 8 is greater than 7. Why? Didn't print this one. It printed that one. Why? Because x and y are not equals, but x is greater than y. But since both of them need to be true in order to be executed, it went into this one. X is greater than y, true. Do this. Since I already did this statement, I can ignore that one. Okay? And this is how you can combine. You can say if, else if, else if, else if, else if, else if, else if. Let's do this. Now you do it. I want to write me a very simple program that checks whether I have an A, a B, a C, a D, or an E as my argument. So what I want to do, I want to execute my program. I'm putting A, and I want to see whether I would have an A, a B, a C, or D, or E. And and the function that you can use is letters. You have seen that already. Okay? So these are my letters. That's a variable defined by R already. So if I say letters 1, index 1 of my letters variable, it will give me an A. If I say 2, it will give me a B, right? So I can use this in an if and else statement. Use this variable to compare to the argument that I pass. Okay, I think we can go to lunch and you can do it after lunch. Okay, just think about it during lunch. How do I say whether an A is an A or A is an B or C or D? Okay?